Welcome back, everybody. Paul and Jay, thanks for joining us. And Ray will join us since we figure out some of the technical back end there. But thank you. I send you the virtual and the physical applause for your talks, and thank you. So panel or participants, please feel free to put your questions in the Q&A. But let me kick off. Both of you are looking at microfluidics in some very different ways. I think there is a conception, my personal opinion, that there's a conception that when we talk about microfluidics, there's a perception that it's just like microelectronics, that we can just go and have common standard ways to fabricate small pipes in plastic or small wells in plastic and have the same support infrastructure to do that we do for microelectronics. I was wondering if you could just comment from both of your perspectives, the fabrication tools that are available to make it easy for you to explore the types of microfluidic devices that you can bring point of care to the bedside. And maybe I'll put one of you in the spot. I'll go with Paul first, since you were speaking first.
Sure. Well, I really appreciate this question. I think in many ways the analogy between microfluidics and microelectronics is apt, but there are serious limitations. One way I think about the limitations is in terms of the charge carriers. So on microelectronics, the electrons are the charge carriers. And because that's so universal across devices, it really facilitates the standardization of design and fabrication technologies. In microfluidics, for life science in particular, the metaphorical charge carriers are often molecules and cells which have their own properties and their own requirements for handling that go beyond the requirements of the assay or the processing steps. And in my experience, that's often a really profound challenge to standardizing design and manufacture. So I think that's one of the limitations of that analogy. Now, in terms of the last part of your question, translating these technologies into healthcare, I think microfluidics has a lot to offer. And that brings us back to, I think, the strengths of that analogy between microfluidics and microelectronics in terms of doing more with less and advancing that capability over time through the miniaturization and being able to handle a large number of samples with a great deal of economy and efficiency. So I'll leave it there, and I'm really curious what Jay's view on this topic is.
Thank you, Paul. I actually think that, when we started this field about 20 some years ago, it was such a far distance between what was even then microfluidics as a field and then microelectronics as a field. But then more recently, I'm actually starting to see a lot more similarity between the two. And the main innovative power behind this microelectronics as a technology is two-angle, which is integration, and then massive parallelization, which combined enable all kinds of amazing devices that we all use every day. And even in the field of microfluidics, I see both happening, not right now. So for example, if you look at the companies like NanoString, for example, which is happen to be and I'm relatively familiar with what they're doing. They are having all kinds of different material, different unit processes, and different operation integrated into devices, not as complex as what the microelectronics devices are currently at, but still very complicated and multi-process, multi-material, different ways of assembling things. So I do see a clear evolution happening towards integration of various different materials and processes. At the same time, my own research has been more focusing on massive parallelization of the microfluidics. I'm interested in making not one microfluidic devices, but maybe 10,000s of them, and parallelize them in such a manner that I can enable such a unique operations that previously was not thinkable. So in my own way, I actually see an evolution and maturation of the microfluidics towards almost the same direction that microelectronics field has gone to about 30, 40 years ago.
The follow-up question, does that suggest then that there's enough, I'll call them platform technology providers in terms of companies that provide the building blocks to connect the fluidic to the electronic to the optical, that it's now as a researcher, you can build and rely on these existing platforms?
Yes, so I actually just came back from my first in-person meeting in 2 years. Microtest is the premier field conferences, international conference, gathers the best work of the world in the field of microfluidics. It occurred about 2 weeks ago in California. And if you go to these meetings, you are actually starting to see vendors. And these are not like, there are integrators, there are kind of companies wanting to become a foundry providers. There are small company and even not so small company providing chip fabrication services or interfacing units and all kinds of component providers. So, even from that angle, and this was like a hybrid meeting, so it wasn't really the most attended meeting, but still I was seeing a vibrant ecosystem already emerging around the field of microfluidics. And that this entire field was grossly invigorated with the challenges of COVID-19 pandemic.
And Paul, is that the same sort of the flavor of microfluidic device that you're using or designing?
Well, I what my lab does is mostly different. We are still reliant primarily on custom platforms that we develop in the small clean room we have located adjacent to our laboratory. But it is nice to see microfluidics increasingly used in industry. And one of the things we've had success with in recent years is taking some of those commercial platforms and using them off-label, so to speak, for our own purposes. And so I think as the commercial systems that are sold proliferate and the price points on particularly some of those consumables drop, it increases the potential for the sort of productive repurposing that we're starting to do.
Very good, Paul. Thank you. Ray, thanks for joining us. Sorry that we got started a little bit of getting you connected on the technology in the back end. We've been chatting a little bit about both what Paul and Jay have been doing in the microfluidic space. Let me put a question to you now. When you look at this space of mutual information, you highlighted looking at both information in image data and information embedded in text data. I was wondering if you could comment a little bit in terms of what other data sources from the clinical perspective might be valuable, additionally valuable, or sort of even more valuable if that's were the case for combining the information from multiple disparate types of data sources. What are they, and how does that look that's different than what you've done already?
Yeah, so I think some trend we've seen is I personally did a lot of research on medical images, which are primarily acquired in the inpatient environment, in clinics, and in the cardiology space. That was my PhD research, and I just finished my PhD a few months ago. And one thing I've heard a lot recently is that a lot of those patients, they start to have implantable devices, wearables, where the caregivers can acquire the data in the outpatient environment, in the home environment, and can measure the physiological state of those patients continuously. And in terms of how to utilizing those data to actually take actions and interventions, I think that's something a lot of AI researchers are interested in, because when a patient is in the hospital, it's sort of the routine is already set up, right? We have some lab measurements, we have the imaging data. If something goes wrong, they know how to take actions. But when the patient is out of the hospital, and for a lot of chronic disease patients, we still want to monitor them. And now the challenge is who's going to look at the data, who's going to analyze the data, who's going to take actions. And a lot of, I guess, recent collaborations aim to solve at least one piece of the problems, which is how to analyze the data, because it's really expensive to ask a cardiologist to monitor the data 24 hours, 7 days. But if AI can do some of the jobs, that will really make the process very efficient.
So there's a question here from the audience. Thank you, speakers. But the question is, like, can you imagine individually some interesting ways to combine your various work? So where you're combining getting data from an image or information from an image, which is data, and then also getting it from the microfluidic device, whether it's on the genomic side or on the viral side. Or what might be some interesting combinations where you actually bring together these very disparate types of data in a targeted clinical application? The broad question, but what might be some areas where you're sort of combining microfluidics and imaging? Any question, I know. I mean, in one area, as I see it,
that one at two levels.
Please go ahead.
So, in some of the work in my group, we are combining already microfluidics and imaging. All of the work in the droplet array platform that I spoke about is using imaging as a readout. And what we've done so far is pretty simple imaging, where we score the intensity in a single color to read out the results of like that that viral diagnostic assay I showed. But we can do more with the imaging and collect complex image data, and so we're starting to work on more advanced analyses, including some machine learning. And talking about crossing over between speakers, Jay and I are departmental colleagues in the Department of Biological Engineering, and we've talked a lot about putting our technologies together. So maybe someday Jay will be able to do that even to a greater extent than we have.
Yeah, so one other thing that I would like to mention is that it is actually in looking at the cell specifically, the cell images, especially the bright field images, is a very rich data set. And we are not utilizing all these rich data that is contained in that morphology, or we usually define as a morphological data set. And it is actually very rich in terms of many important phenotypic informations about the cell. So I see that even in our field, there are a lot of papers emerging, even some of them we are publishing as well, just trying to look at the cell using a very simple imaging modality, even bright field, and then extract and parse out key information that is otherwise not easy to obtain, and using it for analyzing and sorting and also phenotyping the cell. And those kind of ideas are especially important for the emerging field of cell and gene therapy and other new modalities of medicine.
Thank you. Ray, were you going to say something there? Sorry.
No, I think those.
Good. So one of the challenges that our keynote speaker put down is really thinking about the novel ways of engaging with the larger set of patients and caregivers. What's the right way to get faster adoption of the technologies that you're developing? What's that pathway to go from where you are now to getting it in the hands of clinicians and people that can use this type of data? Are there things that we should be thinking about now in terms of our education of our engineering students, our science students, and our doctoral or our medical students that would maybe lay the foundation for better understanding of technology or better understanding of clinical needs, and further fertilizing the academic environment with an appreciation of engineering if you're studying medicine and vice versa? Sort of like, what are the barriers that you think educational opportunities could serve to make it easier for you to have the conversations with practicing clinicians that would close that virtuous cycle of need to technology deployment? Whereas all of our educational system is perfect, and it's, or what are the things that in our ecosystem that we should try to figure out how to change to get faster adoption or change the way that people think about the use of the technologies?
So Paul, if you don't mind me speaking first, I believe I mostly talked about the technology platform, which ultimately lead to the surveillance, a better surveillance. And I think especially in the era of global pandemic that we just went through, and then there are a lot of widely recognized but yet to be materialized health crisis that people are talking about, including antimicrobial resistance and other well-known challenges. So in all of these areas, those challenges may not be necessarily curing diseases in the hospital. It's actually much broader than, it's more like a surveillance at the society scale. But if we don't have a strong surveillance platform in our society, then the downstream cost that is involved with these kind of a health crisis is going to be very, very significant. And we all learned that lesson during the last 2-3 years. So I hope that we could, after this COVID-19 pandemic is over, we could have a more rational and quiet discussions about what kind of surveillance tools and mechanisms should be there in place, so that at least we have this emerging pathogens or other things. We may not be able to prevent that from happening, but at least we can have a fast action against it so that we could be better prepared. So that would be probably the biggest wish list personally for me. And I think a lot of people in the areas of infectious disease seem to agree. And this is not an easy problem because it not just involved the medicine, not just involved the technology, it actually have a societal element to it. So it's a discussion that is much wider than that.
Very interesting. Probably a point in time where that conversation can be had in a different way. Ray or Paul, did you want to add to those comments from Jay?
I would just say from my personal experience as a PhD student in computer science at MIT, I definitely feel I feel like we're like of the environment, in a sense that like during our PhD training, we actually talked with practitioners and really understand their clinical needs, as well as what the potential value creation can be. So sometimes we develop a very novel, cool machine learning methodology, but probably it's not really fit the clinical workflow and the environment. So that's something I really feel we can potentially improve and really create sort of an ecosystem that can encourage this type of communication just across the Charles River.
Yeah, I really agree with. I just wanted to agree with what Jay was saying about the public health perspective and doing as much prevention as we can, and what Ray's saying about understanding the context that these technologies are going into. I would just add one other aspect, which is, even once you've identified a need that can deliver real translational benefits for healthcare, and you understand how it really needs to be deployed to make that impact. I think developing commercial models and understanding for technology that's going to be supported by insurance reimbursements, how that can fit and be supported by an organization that can manufacture and sell and support that is its own really significant challenge that goes a little bit beyond what those of us in engineering and our colleagues in medicine focus on. So I'll just add, we'll probably need some help from our colleagues at Sloan to get these technologies all the way through their potential impact.
Very good, thank you. And I think we have time I'll ask one more round the table question. It's a question I asked at the last panel as well. what's the thing that surprised you the most about your research? In terms of an unexpected result, or you didn't get what you predicted, that was that thing. It's like, oh, well, now we have to figure that out. What were some of the surprises along the way, positive or negative? We'll start. Yeah. So Paul, you went first. We'll go around the room that way.
Okay. One relevant to the work I spoke about is this platform with the droplets, and I showed you a couple different stories that we did, and you noticed, I'm sure, the diversity, starting with small molecule screening, and then looking at microbial community assembly, and finally with the viral diagnostic testing. Now, what I didn't tell you is there are a couple other applications, including analysis of mammalian cells that we've developed there. And so the surprise was how versatile that particular platform turned out to be. We were inspired to develop it by the combinatorial small molecule screening problem I started with. And we're surprised every time. We seem to have quite a natural fit with a new application. And so that's one of the things I love about research is, and especially more fundamental creative research, is you never know where it's going to take you. And with amazing colleagues, both my senior colleagues and my junior colleagues, the students at MIT, it's amazing where those ideas can take you.
Thank you, Paul. Ray, what were you surprised by?
A very specific research project we did. We haven't published yet, but related to the talk I gave on the multimodal analysis using mutual information, is that we're analyzing a large set of clinical data from Beth Israel Deaconess Medical Center, where we have their imaging data as well as the lab test data. One thing we knew is that when a heart failure patient exacerbates, there's a chemical in their blood called pro-NT-BNP that will increase. We also know that those patients tend to build fluid in their lungs, and we can usually see that from chest X-ray images. And one thing I really was, I guess, encouraged by this result was that when we use the images and train a machine learning model to recognize the patterns from the images, we found out that a machine learning model can actually predict the level of changes in pro-NT-BNP based on the features in chest X-ray images. So that really told us this broad idea of utilizing multimodal data from medicine could have huge potential, because in a lot of cases when we have missing data, we could potentially use one modality to infer the other types of modalities because they come from the same patient, driven by the same physiological process. So that's something I would say that was a surprise a few years ago to me, and that made me really wanted to pursue this research direction.
Thank you, Ray. And then Jay, what have you been surprised by in the execution of your work?
I have been working on this ideas of concentrating molecules to enhance the detection for about 15 years now, starting around 2005. And when we started this, it was mainly for enabling the detection of low abundance protein biomarker contained in blood serum or other biofluid. And that's where we started. But then over the years, we encumbered many, many different scenarios, and not all of them are actually biomedical or medicine related. For example, I'm very surprised to see how ubiquitous this particular challenge of limited concentration inhibiting, critical detection that affects many, many decision-making points. So this is actually a universal problem, including, even in the cases of water safety or food safety, antimicrobial pathogen detection, and then recently viral detection. So whenever we talk about any kinds of detection, it almost always seems to be boiling down to the limited number of molecules that you are dealing with in a given sample. And so in a way, it's good for us. We constantly finding new applications. But at the same time, it is really, to me, stressing the importance of making enough progress in this area that we are working on so that we can actually enable good detections in the future. So that is probably the most important insight that I have gotten over the years.
Jay, thank you very much. And Paul, Ray, and Jay, thank you again. A round of applause, virtual and physical. Thank you for sharing your words and joining us today. And that concludes our panel number 2.